Hello, this is the third video of the respiration topic, so 3.3 respiration, and it also overlaps with 3.1 because I just like to teach it like that. So we'll come back to 3.1 and it'll knit into all of this topic as well. So this is the third video of 3.3. We've had the first video, which was a bit of an introduction and glycolysis. Second video was the link reaction and the Krebs cycle, and now we're on to the electron transport chain. Or the process of oxidative phosphorylation. So this is what we've been building towards with all those different reactions, because we had a little bit of ATP made along the way, but mainly a lot of the time it was NAD, this new thing that we've not come across yet, but it's a coenzyme, NAD, that was being reduced. So it was in reactions and there was a series of reactions, and at some point the NAD was being reduced. So that happened twice in glycolysis, once in a link reaction, 3 times around the Krebs cycle, but remember there's 2 Krebs cycles for every molecule of glucose, and we'll come back at the end to adding it all up to working out where we get all our ATP from, because by the end of this it will all come together. So we're on the electron transport chain now, which is this part of this diagram. So if you were inside the cell, the glycolysis has taken place in the cytoplasm, then into the link reaction, the pyruvate from the glycolysis moves into the mitochondrial matrix, the link reaction takes place in the mitochondrial matrix, and then the Krebs cycle takes place in the matrix as well. Then we are here now on the inner membrane of the mitochondria. So you've probably been saying for a couple of years now that the inner folds of the mitochondria are useful because it gives a larger surface area for respiration to occur, and this is the bit that you've been talking about without knowing what it was. So along this inner membrane now we have these complexes here, and this stalked particle, it's sometimes called, that has the enzyme that makes the ATP as well, all along that inner membrane. And that is why we have the cristae, so that we can have more of these systems, and the more of those systems there are, the more that more respiration can take place. We also need, like I said before, the Krebs cycle and the link reaction to be going on in here, and glycolysis to be going on out there, so that those things are ready to come and interact with this part here. So the main thing was, like I said, the NAD, and then to a smaller extent as well, but the FAD. So they are coenzymes, or sometimes called cofactors. And what they were doing was being reduced. So some of you are more familiar with this than others, depends on the chemists really, every time we do this. But remember from GCSE, or you know it from chemistry at the minute anyway, that oxidation is loss, reduction is gain. So in all those reactions so far, the NAD and the FAD were oxidising whatever it was that was reacting. So those intermediates were being oxidised by the NAD or the FAD. So the NAD was becoming reduced, and reduction is gain. So in glycolysis, the link reaction of the Krebs cycle, the NAD was gaining electrons. It was taking electrons. And remember, I kept saying as well that reduced NAD is also called NADH. Reduced FAD is actually called FADH2, and that's because, as well as taking the electrons, they do take the hydrogens with it as well. You don't worry too much about all that, but yeah, they're carrying electrons is the main thing here. So after glycolysis, link, Krebs, there will be a lot of reduced NAD and some reduced FAD here in the matrix available, ready for this process to take place. And just to orientate yourselves, the matrix is here, and in textbooks, and also if you watch other people's videos and things as well, this is the way up that the diagram is. So think of this bit as being like inside. So we've got the matrix here, and then the intermembrane space is obviously the gap between the inner and outer membrane. So when things are moving, if they move from one side to another, that's almost like that's being sent out, and that's coming back in. And again, like I said before, just imagine as well that this is happening right along there and all the way along. Okay, so this whole process is called oxidative phosphorylation. And more in the next video. I'll do one last video for this topic and I'll bring a lot of those key terms together and we'll talk about that a little bit more. But this process is oxidative phosphorylation. But the electron transport chain is actually made up just of these complexes here. This stalked particle with the enzyme, this is related to this and it obviously works alongside it. But when we talk about the electron transport chain, we do mean this bit and people forget that sometimes. So we start, we said NAD, the reduced NAD is the important thing. So what happens is we have reduced NAD arriving here with its extra electrons that it picked up in all the previous metabolic pathways. So here we have, and I'm going to write it as NADH for space really, but also you need to be used to it. So I'll write it as NADH, but I'll probably call it reduced NAD quite a bit as well, so we can use those two interchangeably. So reduced NAD arrives here to the first complex. Okay, so I'm saying complex because this is one of those things that's made up of protein and, you know, various other little bits of other things as well. And you don't need to know and describe exactly what this is made up of, but you do need to know that this is a proton pump, and we can refer to these generally as complexes. And one of the phrases that your specification refers to is being able to reference the fact that here we've got a series of proton pumps and electron carriers, but you'll become familiar with that. But we often call these complexes because they are just made up of not just one thing. So this first complex here, now I'll label it as complex 1 because it's sometimes referred to as that. It has reduced NAD here dropping off its electrons. So if something is reduced, it's got extra electrons, it's now being oxidized to NAD. So NAD, which was reduced earlier on, has now been oxidized. So that means that now reduced NAD has reduced this first complex here. And in doing that, what it's done is it's dropped off those 2 electrons. So 2 electrons dropped off, and that is reducing this first complex. So we'll put these 2 electrons, I know it's all pretty little, sorry. In fact, I'm not going to call it complex 1 because it's messy. 2 electrons, so 2E minus, 2 electrons are being dropped off by this cofactor here, this NADH. So NADH is oxidized to NAD. It drops off 2 electrons here, which in turn reduces this first complex. Then those electrons pass to the next one. So if those electrons move from here to here, and I keep going back to this, oxidation is loss, reduction is gain, is helpful to most of you if it doesn't come naturally, is that if those electrons are leaving this complex here, this one will now be oxidized, and this one is now being reduced. So anywhere where the electrons are going is being reduced. So that one's reduced, that one's now oxidized. So you have those 2 electrons again. They move across. And again. So those electrons are moving down along the chain. Then, and I'll come back to something else that's happening in a minute, I'll just add on as well that FADH2, which is reduced FAD, is also going to give its 2 electrons there. Now, NAD, reduced NAD, always comes to the first complex. FAD always comes to the second complex. Actually, there's, as with a lot of things, there's a lot of this that you don't need to know. So just maybe to help you for it to make sense, and also if you watch other people's videos that might be for university or whatever, these electrons, they don't always go to complex 2 from complex 1. These ones can skip from 1 to 3, but you don't need to know that. So this is why I just show you them going along, and you only need to know that they go along. So don't worry about where they're going and exactly where they're skipping and all that sort of thing. Also, there are these things called ubiquinone and cytochrome C. Again, you don't need to know. So if you see those in videos and things, that's fine, but just don't worry about that. So what they are is they are little electron carriers that can physically move within the membrane. The reason I'm telling you this is because you might wonder why electrons are just jumping from one thing to another. They're not actually jumping. There are other little complexes in here that move along the membrane. They're able to move through backwards and forwards. So they shuttle these electrons to the next one. So cytochrome C, ubiquinone, don't learn them. If you see them, that's all they are. They're passing the electrons along. And again, like I said, some diagrams will show the electrons skipping from this one straight to that one and going along. But for our purposes, all you need to know is those electrons are moving along the chain. The reduced NAD does always drop off at complex 1, and the reduced FAD always drops off at complex 2, but then they move along the chain, down along the complexes. So reduced FAD, FADH2 becomes FAD. So FAD has now been oxidized because it's lost its electrons, and complex 2 has now been reduced. Again, now these electrons will move along. And just to keep the diagram nice and clear, I'm not going to show those as like a second set of electrons, but just so you know as well, I'll put them up there. 2E minus. For FAD, again, it's 2 electrons, so they're going the same way as well. Okay. Now, because this NAD is reduced NAD, it's passing its electrons on, it's becoming oxidized, this one's becoming reduced. Then what happens? That one becomes reduced, that one becomes oxidized. They move from there, that one becomes reduced, that one becomes oxidized. The loser always becomes oxidized. Because of that, this is a series of redox reactions. That's a common phrase that you can use and that you'll see used. The electron transport chain has a series of redox reactions, and what that means that reduction and oxidation is taking place, and that goes along the chain. Okay. When the electrons get to the end, they have to go somewhere. If they don't go somewhere, then this one here will stay reduced, and if it stays reduced, it's not in a state to accept the next electrons. Basically, if any electrons stay where they are, then the complex is almost like, quite happy as it is, it's not got the electronegativity balances between the two for the electrons to move along. So the electrons have to move along to allow the next ones to come. So electrons have to skip along the chain. So they have to be removed from this complex here. And that's why we have a final electron acceptor. And the final electron acceptor is something that you've known since you were in year 7 that we need for respiration. What we need for respiration is oxygen. And oxygen plays such a crucial role because it is the final electron acceptor here. So I'll put O2 there, and we'll say that these electrons, so 2E minus, each time one drops off, it's 2 electrons. We'll go there. Now, if you have 2 electrons, you're trying to make H2O. So then you have hydrogen ions, so protons that are in the matrix here, they can become those H's. The oxygen is one of those O's, and then the electrons are taken. So actually, for each oxygen molecule, each O2, you can make 2 H2O because you have the electrons that come along, and then the hydrogens, the protons that come from the matrix in here. So it would be 2 H2O. and this would have to happen twice. It'd have to pick up the electrons and pick up the hydrogens. Okay. And you know this, it's in the basic respiration reaction that you've learned for years. Oxygen is needed and water is a waste product. And that is a big part of that there. Okay, so what we've had is electrons being dropped off, moving along, redox reactions, and then those electrons being accepted at the end. So the whole point of this is supposed to be to make ATP. So I'll show you how that happens. Basically, every time an electron or a pair of electrons moves through one of these complexes, part of the complex, and the main part of the complex, is that it's a proton pump. So these here are proton pumps. What that means is that hydrogen ions that are in here, so H+, and you just have to accept for now they are there in the matrix, there, bobbing around. They will be pumped through those complexes every time the electrons move through. And you can think of it in different ways. You don't have to be able to explain this, but you can think of it as the electrons moving through almost gives it a little bit of energy to pump the protons through, because the proton pump will only pump protons when the electron's moving through. This second one isn't a proton pump, so I'll just... So the hydrogen ions, these protons, are being pumped through, and they're only pumped through as the electrons pass through each of those complexes, those proton pumps. So you have to get used to calling these complexes, but also calling them proton pumps as well, because that's their main role here. So just there in the matrix, there are protons, and they're being pumped through. You can also think of it, I said you can think of it like it's giving it a little boost of energy to pump it through. You can also think of it, I'll get that closer there. You can also think of it almost like there's a little bit of negative charge, and that helps the positively charged protons move through. But like I say, you don't have to explain it, it's just so that you understand that that's what's happening. So the whole point of this, the whole point of these reduced NADs, reduced FADs, they've gone through all of that glycolysis, link, Krebs cycle, to bring those electrons. The electrons move through, the protons have moved up. The whole point of this is to build up a concentration of protons out here. And when I say out here, remember I actually mean in here. So we've got lots of hydrogen ions, lots of little protons building up there. What we get is a concentration gradient. So we get, because this is taking place over and over and over again, we get a high number of these protons out here compared to in there. We get a concentration gradient building up. And what happens is, once this has taken place enough times, and remember, not just here, but all over that membrane, once it's taken place enough in a localised area of that mitochondrion, there'll be enough hydrogen ions that the gradient can be released, and that release of that gradient generates energy to make ATP. So this is where we come to this stalked particle. Now, nearly everything that I've read calls it ATP synthase, but WJC call it ATP synthetase. So here we go, we'll call it that. So I'll write it here. ATP synthase. There we are. There. So ATP synthetase, and that's the enzyme that is associated with this electron transport chain. There's a massive build up here. We've got a big concentration gradient. And what happens is it's like a floodgate opens, and the hydrogen ions are going to be allowed through there. Now it is you can think of it like build up of water at a dam, and then a water wheel allowing the water through, which generates energy as it spins. And that's not just me. so I need to remember it. That is the analogy that's used for this thing. Because what actually happens is, this part here does really spin. It actually, it's weird, it looks like a machine, and it spins round as the hydrogen ions go through. So it is a very useful way to picture it. So there's a build-up of protons. They're allowed to move through. It's like a whoosh of them all moving through. That energy allows it to spin. It allows it to use that energy and harness that energy to make ATP. And if you were thinking of it being like water, you imagine water, you've got, if you had a high concentration of water out here compared to in there, and it moved through a membrane, you would call that osmosis. It's not osmosis because it's not water, it's protons. So because it's a chemical charge, it's a different thing from water, we call it chemiosmosis or chemiosmosis. So just think of it in the same way as osmosis. It's a build-up that has to move down its concentration gradient, but it's called chemiosmosis or chemiosmosis, and it goes through. So those protons rush through there, and what that does is it generates ATP. So we have ADP, adenosine diphosphate, and obviously an inorganic phosphate ion. So you need an inorganic phosphate ion added to ADP. I don't think I wrote that in earlier on, but we'll talk about that. In Krebs and all that lot. I just wrote ADP, didn't I? But ADP and inorganic phosphate, and then that will be converted to ATP. And that's the main way that ATP is formed in respiration. And that whole thing is called oxidative phosphorylation. And it's called oxidative phosphorylation because we've got phosphorylation taking place here. A phosphate has been added. It's gone from di to tri, so a phosphate has been added. And it's oxidative because it requires oxygen for that process to take place. Okay, so oxidative phosphorylation. And we'll come back to all these other key terms, like I said before. Okay, so when this takes place, when the reduced NAD and the reduced FAD pass on their electrons, the gradient's built up, then it's released, the hydrogen ions rush through, the protons rush through, and the ATP is made. It's not just one ATP. So what I'm going to do next in a moment is show you how we tally up how many ATP are actually formed from this process taking place. So I'll pop some hydrogen ions in there. They're the ones that have come through. Now obviously because they come back through into the matrix, they're now here ready to go through again and be pumped again by the proton pumps and for the whole process to continue. So it's just happening all the time. As long as glycolysis and the link reaction and the Krebs cycle are all taking place, we'll have these reduced parts here, and that will mean that this can take place and then the ATP can be formed. Okay. So what I'll do actually, I'll do a separate video where I will add up the ATP, and then we will look at some of the key terms in that one as well.